chapter 14. So this is the second chapter for exam 4. So we started with chapter 12. Now we're moving to chapter 14, but we'll go back to chapter 13 for the end of your exam 4 material. So exam 4, chapter 12, chapter 14, then chapter 13. So for chapter 14, we're focusing on the brain. So for our central nervous system. So we're going to look at the major parts of that and look at the major landmarks. For our brain, we have two different directions. So rostral is going towards the anterior or front portion. That's just another directional term. So a way to remember this is rostral, nostril. So towards like the front of your nose where your nostril is. Caudal is going in the opposite direction, so this is our posterior direction. So our main pieces of our brain, we have the big chunky section of our cerebrum, the bottom really wrinkly portion of the cerebellum, and then exiting out of that foramen magnum of the bottom of our skull, we have our brain stem. So that's all the pieces of our brain, and then that switches, once you leave the brain stem, to our spinal cord. So major landmarks, our brain has two halves. So separating our two halves, we have our longitudinal fissure. So that separates our brain into the right and left hemispheres. In order to communicate between the left and right side of our brain, we have the corpus callosum. So that's this bridge here in orange. It's made of white matter, and that allows us to communicate between our two halves. Going back to our exterior surface, we have what makes up the wrinkles. So the cracks in the wrinkles are our sulci, and then the peaks of the cracks are the gyri. So you'll also see this as sulcus for one, gyrus for one. So the other, what's there is plural, yay for Latin. Okay, I'm also going to show you the medial aspect of the brain. So we have a mid-sagittal cut through our brain. So here's our corpus callosum again. And I'm going to point out certain little features that you'll hear me talk about. You have your pituitary gland. This is your major hormone control center of the body, controlled by your hypothalamus. Then again, your brainstem here. Your brain has multiple lobes, just like your liver and your lungs. So there are very deep sulci, so in this case one sulcus, that separate the different sections. So for example, this central sulcus separates the parietal lobe and the frontal lobe. Back here we have the parieto-occipital sulcus that separates our parietal lobe and occipital lobe. And this is just a cut through of our cerebellum, where you can actually see a little bit of white matter there too, called the tree of life, or arbor vitae if you've heard that. So it kind of looks like a tree. We'll talk about that later. Okay, so if we took our brain and made a different type of cut, so in this case being a coronal or frontal cut through the brain, we're able to see that we have two different colors in our brain. So we have white matter and gray matter. So the gray matter portion has very little myelin, so that's why it has a darker color. The myelin gives kind of a white appearance because it's mostly fat. So gray matter, that is the neurosomas, our main cell bodies, our dendrites, and synapses that are located there. Our white matter, which is found deep in our brain and superficial in the spinal cord, has lots of myelin. So that fatty tissue creates the white coloring in those areas. So our white matter allows us to communicate between different portions of the brain, while we'll actually make connections and make those synapses on areas, for example, like our cortex, which is the outer gray matter portion. And then we have some nuclei that are hanging out in the center of that. So let's look at our brain versus our spinal cord. So in our brain, again, the white matter is on the inside, so that's our myelinated axons communicating to different portions of our brain. So they're able to send messages along. The gray matter has your neurosoma as well as your dendrites. Spinal cord. Gray matter's on the inside. So here we have our neurosoma, but then the axons are leaving that central portion of our spinal cord. Why? So it can go target something. So here, this axon is going to eventually lead to a target cell, and they're able to communicate to whatever your target is. So it could be another neurosoma, well, neuron, or it could be a muscle cell. So we're going to get back later to all the functions of the neurons, but now we're going to look at something that covers on the outside of the brain, which is our meninges, and different chambers that located on the inside of our brain, which are called our ventricles. Inside our ventricles and some of our meninges, we have a fluid that goes around there called the cerebrospinal fluid. And in this area, you also have ways to protect your brain, so creating your brain barrier system. So let's look at meninges first. So you have 3 layers of tissue around your meninges. The 3 layers are called the dura mater, the arachnoid mater, and pia mater. You might not see this when we do our dissection, because sometimes these get ripped off, even by accident. But their job is just creating a cushioning layer in between your skull and your brain. There's also some of that cerebrospinal fluid that flows in that arachnoid mater layer. So in this space called the subarachnoid space, that is where that fluid flows. Let's look at these ventricles. So these are chambers inside your brain, and that is where our cerebrospinal fluid is created. So our ventricles are 4 internal chambers. At the bottom of each ventricle, we have the choroid plexus. So there's a bunch of blood capillaries that are creating this choroid plexus that's at the bottom of the ventricles. Inside that area, we also have our ependymal cells, or ependyma, which is a type of neuroglia, helper neuron type of cell, so our helper nervous system cell. So it's those cells that are actually going to produce the cerebral spinal fluid. So this cerebral spinal fluid fills your ventricles, as well as different canals that connect our different chambers through our central nervous system. It's going to eventually exit out and bathe the external surface of our brain, so in that subarachnoid space. And it also goes down into our spinal cord. So our cerebrospinal fluid, produced by the ependymal cells, is created through the filtration of blood plasma. So those capillaries in the choroid plexus is where it's filtering out that blood and just taking the liquid portion of blood instead of all the solid bits. So like your cells and platelets. So with our cerebrospinal fluid, it has one directional flow that it goes. So we start, and each of our ventricles will be able to produce fluid, but we're going to start at the furthest locations. So number 1 and 2 are the lateral ventricles. So I'm going to go back to our little picture earlier. So our lateral ventricles are these 2 here. So they're on the lateral or outside of midline. So those 2 ventricles there. It then moves into the number 3 location, which is the 3rd ventricle in the middle, going down to the 4th ventricle, filling our subarachnoid space, then being reabsorbed by the arachnoid layer, and then finally being returned to the bloodstream. So after it's done its circulating, it's the same fluid that came for your bloodstream, it's going to return to your bloodstream. Here's a little pictorial version that's probably a little easier to see. So 1 and 2 lateral ventricles to the 3rd, to the 4th, subarachnoid space. So this is our first stop after we leave the ventricles. Then are reabsorbed by the arachnoid space and returned to the bloodstream. So that's the order I want you to know for that question on your study guide, because it's on the test. Also on the test, so with our cerebrospinal fluid, what does it do? So it helps with buoyancy, it helps our brain float, so it just kind of gives a good cushion. So instead of our brain succumbing to gravity and just resting on the bottom of our skull, it floats in our head. So this is why when you have an accident and the brain jiggles around in your skull, it's because it's just floating in that space. Next thing is it provides a cushion for your brain, so a layer of protection. And then also works as chemical stability, so literally a brainwash. So as it's moving through your brain and over the surface of your brain, it's able to rinse out some of the waste, and then that can be returned to your bloodstream so that your kidneys can then get rid of that waste. Moving on to our blood supply. So our brain needs constant oxygen as well as constant glucose to make ATP to be able to feed its cells, so it has to have good blood supply. However, we don't want everything that's in our blood, especially if we're having an infection, to be able to get across. So that's why we have our brain barrier system. Our brain barrier system is created by two different sections. So two different possible entry points into our central nervous system. So the first one is our brain tissue capillaries that are just running throughout our brain tissue. So just getting to all of our different neurons. So that blood-brain barrier, which is what we've known for a long time, is created by the perivascular feet of your astrocytes. And I'll show you a picture of that again. The other type is the capillaries in the choroid plexus. So what we just talked about just a second ago, the ones that are making up the bottom of those ventricles where we're creating our cerebrospinal fluid. So they're creating the blood cerebral spinal fluid barrier. So these are two kind of entry points that have to be guarded. So our blood-brain barrier system is like bouncers at the door of Club Brain. Yeah, no, that was cheesy. Oh well. Okay, so blood-brain barrier, tight junctions between endothelial cells, so you do have those cells that create the capillaries. But mainly what I want you to know, perivascular feet of the astrocytes create your blood-brain barrier. For the other barrier, blood cerebrospinal fluid barrier, so our ependymal cells, there's tight junctions between it, and that helps to keep stuff from getting into your brain. So what can get in? So going back to our nightclub type of reference, so stuff that gets a VIP pass. Water, our cells need water, so that's easily able to get in. Plus you have the aquaporins that are the water channels that allow for water to move in and out cells anyway. You also have glucose. Glucose has its own channel. Plus your neurons, they need the glucose in order to make ATP, so you need to make sure you have enough glucose. Last thing is anything that is lipid soluble. So that's able to go across that plasma membrane phospholipid bilayer in order to get into the cell. So stuff that is lipid soluble, we have oxygen, so we need that nutrient. Getting rid of our carbon dioxide waste product, so that should go easily. Other things that are lipid soluble, you have alcohol, caffeine, nicotine, anesthetics, so all things that easily affect the brain. That's why you get, have the effects on your brain when you get drunk with alcohol. Caffeine wakes you up, easy. Nicotine, you get that buzz from that. Anesthetics, get knocked out. Well, for a short time. And then that wears off and then you wake up. Okay. Some other stuff has regular entry, so it's somewhat regulated. For example, our different ions, sodium, potassium, and chloride, waste products that are created by the cell. There is some regulation of getting that in and out, but it's still able to be passed. What cannot pass, or it has a very hard time, is stuff that's rejected that is highly charged. So again, going back to our phospholipid bilayer, because of that type of molecule, it would be hydrophilic. So it's likes water over something that's lipid, so it would have a hard time to pass through that plasma membrane. Also, anything that's very large, so it wouldn't be able to leak past the gaps that you do have, some of them, in your capillaries that aren't covered by perivascular feet. So You can have a little gap space, but if it's too big, then it's not going to get across. So sometimes we have psychotic medications or other medications that need to be crossing into the brain and have that effect, but we create it in a petri dish and it works, but then trying to get it past this brain barrier system is too big. And so the drug that technically would work, we can't get it past that brain barrier system. So that's kind of the reason why sometimes when you're getting medications for certain brain conditions, that it's kind of hit and miss, as you kind of figure out, okay, what can get across, what your body's allowing, so we can actually treat you. I'm going to stop here for this first section of chapter 15 because we're moving on to a different subject.